this? Or?
Yeah, we'll just start by acknowledging the traditional owners of the land on which we meet today, the Turrbal and Yagara people.
Okay, welcome to the talk today. It's a short notice, but thank you for coming. My name is Ross Darnell. I work at CSIRO, and about 15 years ago, we collaborated with people from Keio University, and then had a joint project work through the Surf Hub project, which was back in the 2009-10. And ever since then, we've had connections with Professor Shimadzu, and I'd like to introduce him now. And he's from Kitasato University. He used to be with Geoscience Australia, St Andrews, Loughborough, and now he's with Kitasato as Professor of Data Science. Thank you.
Thank you very much. Thank you very much, once done, Nivea, hosting and organizing this seminar in short notice, and everyone, thank you very much for coming. I'm very much excited learning from you. I'm very much capable of saying a little bit lying things, but hopefully you get point me out and give me a little bit that right direction for me. And yes, so today my talk is about biodiversity stuff, and I'm quite sure I don't need to explain things for you. So biodiversity stuff, that's something I've been working on. At the same time, I've been struggling, and hopefully I can get a little bit light from you guys today. So biodiversity stuff is pretty much big topic these days, and well, not everyone, but many people talk about. And particularly United Nations pushing kind of direction, which is quite good for this center, I imagine. So the last decade was a decade of biodiversity, so try to promote the concept of biodiversity. And now we're having decade of ecosystem restoration. So we need to somehow make nature positive by 2030. And this is a quite iconic figure, just sending out strong message. Yes, we must do something and make it positive. And my cynical way to look at this one is, okay, what's the y-axis then? So that's been the kind of my question a long time, and this is something I like to share with you as my pain. So to measure biodiversity or quantify biodiversity is a little bit tricky business, as long as I understand. And I tried hard what actually people measuring and what they're talking about biodiversity. And this is one of great paper published some time ago, giving us some summary of biodiversity indices back in that time. And this is only one table, and I think this paper got more 2 tables to go. For risk of indices basically. And it's not everything, of course. And since then, it's more than 10 years. I'm quite sure the number of indices are exploding. So having my small brain, actually I can't cope with this number of indices, what that means. And of course, if you look at one by one, and if you choose carefully which one you want to use, that's making sense, and it's got their own right to be used. And typical measures like richness, abundance, these kind of things, and the hill numbers. So this variety, yes, quite nice to see this kind of active research going on. But sometimes I feel like a biodiversity index itself defines biodiversity, rather than the concept of biodiversity defines what the biodiversity is. So I'm trying to understand what actually biodiversity means in a quantitative sense, because I've got my background is more statistics background and also Japanese background. Trying to figure out what actually we're doing. And for the last decade or so, while working on this topic, I've sort of felt some gaps in this biodiversity study field. And as you know, biodiversity is about community ecology topic. So basically, you're talking about ecological communities. And underneath, there's a legacy there in ecology, population ecology. So you can come across lots of giants name who worked or proposed a theory in this field, basically looking at population. So basically the population means the group of the individual belongs to the same species. So in other words, a community was basically consists of these populations. And of course, population is consists of individuals. And on the other hand, having advanced technology these days, we got more data stored, we got more power for the computation. Species distribution models are quite a common tool to investigate what's going on. And this is a good example talking about species presence absence, taking account various environmental factors and variables. And you can get nice maps. And this is good. And people working, basically when you build a species distribution models, what you're doing is basically identifying the probability distribution, what this presence, absence, or what abundance follows. So basically identifying, if I write probability distributions function F, then this is what you identify by doing species distribution models. And this got pretty much popular these days, and I'm quite sure many of you is already using it in some extent. And it's another area, mathematical ecology. It's got its own deep history behind it, and talking about population dynamics, how they operate, or how the abundance size changes over time, something like that. So usually described by differential equation and something like that. And what I'm feeling or what I'm thinking is people working in this field, ecology field, and people working in data analysis modeling, spatial distribution modeling in this field, and mathematical biologists, we don't talk to each other that much than we should do. So the kind of missing communication makes a gap among our research. That's a kind of a little bit well, saying is easy and doing is difficult all the time. But I feel a little bit is there anything what I can do? That sort of feeling. And it will be great if we combine, in a sense, or integrate this already established legacy discipline under the name of biodiversity study or research. And I thought about actually having this kind of lots of gaps, what we actually missing from this. If I summarize it sort of two direction, one is this one. So the vertical axis try to represent, so here in ecological community, it's got the hierarchical because the community consists of collection of population and each population consists of each individual. So it's got the hierarchical and additive structure in it. But if you're only working on population ecology, you don't talk about community ecology. So this hierarchical structure is one of the big gap if you don't really cross the, well, I wouldn't say discipline, but cross the field. And second axis I'd like to put is some stochastic and statistical nature of community or population dynamics. So usually this species distribution model gives you a map, which is snapshot. So the state of a time given or you decided. There's no dynamics there. Mathematics, mathematical biology or ecology, they're quite good at dealing with dynamics. because of the nature of differential equation real. But this is well, these are quite deterministic expression. Of course, these days you can use stochastic differential equation type of approach, but popularity is still a deterministic world because it's easy to understand. And again, because of technology, mathematics, sometimes technology people try not to get involved. So these two directions is a kind of big loss if we don't communicate each other and try to do an integrated research. And I wonder if I could do something that will contribute this direction. So this is my focus or focus of this topic today. So before jumping into the topic, let me a little bit explain about how I understand the common or famous concept alpha, beta, gamma diversities. And I know there are lots of arguments and discussions still going on, but I'm not going to that direction. Just I show my sort of ignorance in a way. This is how I understand. And please tell me how you think or how I'm wrong. And then I'm moving into the way how I try to understand biodiversity concept from here. So there are three common class of biodiversities here. So it's called alpha, beta, gamma. The alpha is, as long as I understood, that's a given the community and how diverse within the community. So it's often called an average within the community diversities within. And beta is basically you need at least two communities and compare both. So if it's kind of spatial context, you're comparing difference. If you're comparing temporal context, and then it's basically if I say time t and time t plus 1, then you are looking at a change between two occasions, but same community. That's it's called beta diversity, so between community diversity. And if you talk about the whole, this community as a whole, then that's called gamma diversity. Then the question is always, how are you going to measure it? And if you use certain measure like a Jakarta, something like that, and then you can, for sure, you can calculate numbers, you can get numbers. But my question is, why that numbers matter? And why this specific index? We need to look at. But Whitaker's idea is something like, as long as I understood, is it sort of in line with your understanding? Am I not that far? Good, thank you very much. So what I'm going to do here now is a little bit more think about alpha diversity, what this actually represents to us. So as I said, alpha diversity is basically the within community diversity. So that means I would say sort of well, if you think about sort of evenness, so basically here example. So here is a community A, it's got one individual, and here community B, it's got 3 individuals for each species. So in terms of evenness, the same. But if you have like this, then evenness differs. So basically, it's got scallop got 3 individuals, dogs got 4 individuals, and turtle got 2 individuals. So it's not even. So basically, how differ from evenness is something talking about alpha diversity. So that's my interpretation. Is that something you agree upon, or you get sort of completely different idea? So I think that's a sort of common understanding as long as understood. And beta diversity, so as I said, between community diversity, so it's temporal context is called change. And I would call it difference from the community to another. So if you think about location A and location B, and if you move, and the question is whether you see any difference between these locations. And if you find the same, exactly same, like 2 kangaroos, 3 koalas, and 1 emu, let's say. And if you find another place the same, then probably you wouldn't recognize the difference between the community. So that means basically no change or no difference. So how change or how different happened between 2 communities that measures biodiversity or data diversity. So that's how far from similarity. So that's my understanding. And if I convert that idea into statistics context or probability context, I can write the concept of evenness and similarity, something like this. So first one, evenness, which I talked about for alpha diversity, it's basically assuming that the probability distribution for species abundance. So let's think about abundance here. N1 is the number of individual abundance for species 1 at area A, and N2 represent number of individual for species 2. And what this first line says or states is basically the probability of getting number of individual for each species, the same location, is the same. In other words, that infers the mean value for the abundance, number of individuals, the same. So that's making sense because that's evenness. So we expect emu, kangaroo, what else? Koalas, their expected abundance is the same, even. And second one is a similarity, which is slightly different from evenness in terms of mathematical description. Here, what I'm writing here is these bold font NT represent the basic vector like here. So as a collection of abundances and at the location or time point t, the probability of the just basically joint probability of abundances are the same whether you have time t plus 1. And that also infers in terms of the expectation is the same. So in statistics, often we call it sort of stationarity assumption, if you're familiar with this kind of concept. So this is my sort of starting point, how I'm going to describe evenness and similarity.
You don't really care whether it's time or space, right? Like location.
Yeah, for now, I'm talking about a little bit time, but you can replace this as a A or A dash or something. You can use that. And pretty much the same concept. Yeah, thank you very much. Yep. So this is my starting point. And then from here, what I like to say here is actually the biodiversity measure like alpha, beta is something like I can describe. So let's assume we got the joint probability distribution of abundance for time t, like f and t here, and theta t is a parameter that defines the probability distribution, joint probability distribution. And then one time step ahead, the joint probability changes or the same, like f and t plus 1. So that's the kind of basic notation. And if we think about the whole community, that would be specify the joint probability of time t and time t plus 1 like this. And you have more parameters to define. Using this notation, the left-hand side, this is basically the whole community probability joint probability distribution can be dissected into 3 components in multiplicative way. First one is basically taking a probability ratio between the whole community and sort of reference probability distribution but that basically describes how probability changed from this point, this distribution to the current joint probability distribution. This is a ratio. And second part is trying to represent evenness. So if you assume sort of the same probability for each species, and then this reference point, the ratio again, describe the difference between, or difference from the evenness. So that is the divergence from the evenness. And here, this is basically the sort of origin point when you try to compare species communities. It can be uniform, but it's not necessarily uniform, but something like that, sort of quite standard probability distribution you can show. So what I'm framing here is alpha, beta, and diversity can be formulated in a multiplicative way, just multiplying, just for the easiness, let me call this as a uniform. It's not necessarily uniform, but uniform, times beta, alpha, beta, then current, basically the community as a whole. And the equation itself is quite trivial. I'm doing nothing, to be honest. First instance, some of you find it, what are you talking about? It's too obvious, but this is what it is. And in fact, if I start taking this probability ratio, let's assume, okay, let me talk about a little bit focusing on beta biodiversity rather than alpha, so the green part. So let me set the reference point, say theta t as a, theta b as a theta t, so that's basically the time t point, the parameter. And then using conditional distribution, I can rewrite a joint probability distribution like this, and then actually this part cancels out. So basically what you're looking at is conditional probability distribution. And let's assume number of individuals follow the Poisson distribution, so one of exponential family distributions. I'm going to use this notation, which is probably different from what you're familiar with as the Poisson distribution, but believe me, this is a Poisson distribution. And if I use this notation, it becomes quite nicely written. So this beta diversity part becomes exponent to the linear combination of number of individual times parameter alpha. Alpha is a ratio, log ratio between expected abundance of each species at time t and time t plus 1. And then this kappa part is sometimes called cumulant function, which is unique to the Poisson distribution. It takes something like this form. And this part is not the but the important part is next one. So taking all the expectation of it, I show you the key part. What happens is, assuming a Poisson distribution, at the end, I get this formula, quite simple formula. So let me clarify it again, notation. Lambda JT is the expected number of individuals for J species at time t. And JT plus 1 is the expected number of individual J species at time t plus 1. And alpha J, so that 1 plus alpha J is a multiplicative factor, multiplier, to the time t expected number of individuals. And then you're going to sum them up from 1 to S if you have S species in the community. And does this form ring a bell? It's quite simple formula. And I'm quite sure you came across this expression when you did ecological theory or theoretical ecology class a little bit. So let me come back to the point I made before. So what I'm thinking was, basically we are missing two directions. One is stochastic statistical nature of community population dynamics. If I rewrite this formula, focusing on species J, I can rewrite this way. So the basically beta diversity part is minimizing alpha J if there is no change. If there is no change, that means the number of individuals never changes. And this is a population dynamics model, the simplest one. And if you solve it, it becomes just exponential increase, something like that. So here, what I did is using stats concept, taking into account probability distributions and all kind of that, using expected value, I connected this direction. So now it becomes population dynamics model in terms of average. And what's the next bit? So hierarchical additive structure. If you look at this part, this is basically summing up whole species, each species, additive structure there. So it's quite simple once you start from the idea what the alpha beta meant to be. Well, at least for me, that's more making sense to me now, what actually I'm trying to quantify it. And I'm confident I can speak a little more what beta biodiversity means. So what I tried was basically try to get a more integrated way to express what beta diversity here means. And it comes together nicely, I think. So up to this stage, is there any question you want to ask or anything point I made a mistake or said lie or nothing? Okay, very good. Thank you very much. So this is a kind of theory I thought about, and then I felt like, okay, so that's very good. It turned out quite simple expression and easy to understand. So let me move on to a little bit example of how I'm going to work this framework is. So coming back to the idea of population dynamics, if you look at one species, sorry, it's now not described as a I other than J, sorry for the confusion, but basically the instantaneous increase rate alpha can be described this way. So if you're doing a little bit finance and something like that, so this is exactly the same as you see how much you gain, something like that. And then if I sum them up together as a whole community dynamics, what I need is just simply sum them up because of the hierarchical structure, additive structure. And then I know what the instantaneous change rate of the community. Easy. So if I use this framework, what I can do is basically I can get more information from beta diversity, the data I analyze. So what I'm going to show you as an example here is basically a time series record of A communities. So that if I draw a picture, this is like this. So basically having abundance on y-axis, we have observation over time. And then try to understand what the beta diversity, or sometimes people call it temporal turnover for this community. So the good things about the framework is basically it can be decomposed in two key concepts. One is basically the measuring the change in composition, basically species composition in a community. And the other one is a change in community size. And again, these components here, D1, D2, is additive. What I'm trying to say here is I'm not going to too much detail about this equation, but I'd actually show you some output example. If you use, for example, GLM, generalized linear model, to see the species distribution in modeling, what happens is actually, if you take into account the species, and then you can tell which species actually driving the change, like ANOVA. And that contribution is additive. So each species additively contribute to the change. And if you use environment factor in your model, in GLM context, your environment factor contribution is also additive, becomes additive, like this. So that means you can easily visualize which environment factor is actually driving this community change, or which species is actually changing the community or contributing most in the community change. So here's the example. So I've got the colleague who've been working or collecting fish at Hinkley Point here near the power station, and he collects over 40 years by now, I suppose. Every month, he collects the fish community and count the numbers and identify species. It's quite impressive work, and I had a privilege to share the data with him and analyze the data. So what I'm going to do is use that framework to extract the change of biodiversity, peak diversity, to see what's happening with this fish community. So here's one example talking about changing composition, green part, and second part changing in community size. So for now, just you can ignore the red part. So if you look at green part first, so that is basically a change in composition. So if there is no change, it's supposed to be 0. But it's getting far from 0 as time goes. So that means basically this species composition is changing since 1980. And if you look at changing community size, blue line, it's fluctuates, of course, but the tendency is basically increasing. So that tells you that the community size is increasing here. But of course, this is a sample, so whether the sample represents well the local community or not is another issue in terms of sampling. But at least in terms of reading what this means is something like that. So relatively simple. And let me show you which species is driving, which environment factor is driving this kind of change. I can easily visualize here. So here, the color shaded area represents basically the size of contribution from a specific species. So here, I highlighted just cod and goby and whiting here. And as you can see, in this community, cod is a main driver. This one's playing big role in this community. Whereas goby, it used to be big player, but as time goes, it shrinked. So that means the influence of goby for this community is getting small. But whiting is slightly increasing. So these kind of things we can extract from the biodiversity concept. And once you know which species is playing some important role, then you can look into more detail what's actually happening in this community. So rather than just looking at numbers, it still gives you kind of idea what we need to look more carefully. And next example is environment factor. So I didn't have many environment factors for this time, but if you have 100 or more, you can use exactly the same, but just for the sake of illustration, I use the 3 variables here. One is EI effect, which means pretty much nothing, but tide height and water temperature. So that tells me basically tide played a quite important role in terms of community change. But that making sense. So this is something quite useful feature from the framework we talked about. So I promised the 30 minutes and 5 minutes or so, a little bit, 5 minutes each, a little bit behind, sorry for that. So let me summarize my talk. So I try hard to understand what biodiversity, what alphabet of biodiversity means. And my so far conclusion is I can formulate it using probability ratio, describing community abundance, mimicking a probability, joint probability distribution. And the nice things about framework is it comes down everything additive structure at the end, because that's a part, or mostly because that's the nature of beauties. In the ecological community, there is basically a hierarchical additive structure, which sometimes we ignore, but now we can appreciate that structure plays a quite important role in terms of understanding biodiversity change. And as I showed you, you can identify which species is actually driving the change or which environment factor plays a key role. So that gives you sort of idea when you're in a position, for example, making a policy or executing policy, which one you need to look, the environment factor you need to look up or think very carefully, or which species you need to protect. So hopefully this kind of framework, that helped me understand what biodiversity we're trying to quantify, and also that gives me the useful way to actually use this information rather than just plain numbers. Thank you very much. That's it for today. That's what I wanted to say. Thank you very much.
Well, thank you very much.
Thank you.
The talk.
Although this time is the quite busiest time of the year, I'm quite sure, but thank you very much. I very much appreciate it.
Thank you very much.
you.
And thank you to Emily for organizing for us while Kate is
Pleasure. Thank you.
Thank you.